hopefully today I can convince you some of our newly invented tools will be useful for your study, and we will welcome any collaboration request, just email me. So my lab work on ultra-high resolution imaging through different aspects. And today I'm also going to talk about a little bit more theoretical work on a new theoretical resolution limit. So lab works on 3 different general directions with Maliki's wonderful introductions of super resolution concept and many beautiful images. I don't have to explain further on that concept. But taking super resolution, we want to make it work in live cells. For example, over here showing different clustering collated pits, actually where it's moving during acquisition about 1 minute, and 2-second snapshot, you can see this ring-like structure, 3-second snapshot of focal adhesion, and also these are the acting patches for yeast. We're also moving super resolution from fixed cells close to the cover slip into tissues and hopefully small animals as well. What we demonstrate here is amyloid beta fibrils in 30 micron mouse frontal cortex in Alzheimer's disease models. We built new instrument, the one that's about this big and this long in length, about 1 meter in 1 meter by probably 1.5 meters in sizes, to incorporate new physics and new chemistry potentially to allow higher resolution into the systems and targeting with 1 to 5 nanometer resolution. And for the time limitation for this talk, I will try to focus on this middle section over here to tell you more about how do we do this in tissues. For super resolution imaging, the concept based on single molecule localization. Here we can see a structure which is diffraction limited, and on the right side you can see this conventional image. If you can isolate each individual molecule and localize their centers, you can accumulate their center in the super resolution image, localize single molecule and accumulate their center in super resolution image, therefore you build a structure. However, this movie only shows you how to do it in 2D, but how do we make it in 3D? Traditional 3D super resolution imaging method based on biplane, astigmatism, double helix, and phase ramp share a similarity. What they do is they try to put in some optical element such that the point spread function will change shape at different Z positions. Showing up over here, you have a point spread function in focus, which is round. If you're out of focus, elongate in one direction, and all the other direction in Z, you will elongate in the other direction. If you look at these type of data obtained in the lab, 3 different setups, you have astigmatism, and you have biplane setup, you have 4-pi single molecule setup. Look at their blinking data, you can find out some of the molecule elongated in one direction or the other. These elongation in different directions tell you the Z information from where the molecule is originated within the cell or tissue. Biplane image 2 different axially separated planes, and you can see their differences between their focuses of 2 planes to decipher where they are. There's more interesting data showing up over here based on 4-pi single molecule imaging. Basically, we combine 2 objectives and coherently detect the single molecule fluorescence, and you can see their patterns are a little bit complex. They're in general astigmatic, sometimes elongated in one direction or the other, but inside them, they also form interference patterns. And these are single molecule interference patterns formed in a coherent detection of these are their events. And they encode much more resolution, much more information per photon count. If you look at in general what this looks like in terms of their point spread function, astigmatism give you different elongations in different Z, biplane different give you different defocus at different Z. Is 4-pi point spread function give you different interference patterns and also different general shape. To decipher where your molecule is, you're basically given a picture of your detection on the camera. How do we infer where they are in cell or tissues? The general direction or general calibration method for this type of approach or 3-dimensional single molecule is basically put a bead on a cover slip. You move your piezos up and down, trying to find out how they look like at different Z. For example, in focus over here, you can get an in focus point spread function. Going down, you get an out of focus point spread function. And going up, you get another out of focus point spread function. So this gives you a relationship of where the molecule is in your cells at different Z positions, and what's the width in Y and what's the width in X. You basically take one of your single molecule patterns, measure the width in X and Y. You do this lookup table and look at, oh, it's actually 400 nanometers. And this is how you find the Z. This is a traditional method, has been used for a very long time. Even in single molecule, before super resolution is invented, this has been the traditional method. This method actually has some caveats. The photons emitted from your calibration samples, for example, the bead, actually never goes through your sample. No matter what you put on top of the cover slip, right? You can put tissue down there, you can put cells down there. The photon go directly to the objective. It's not affected by your sample. However, what we really image in the tissues or cells, those photons that you detect on your camera has to go through the sample first. And this is what we call the in situ PSF. And therefore, there actually is no method before the work I'm going to talk about. There's to retrieve this in situ PSF. If you don't know how the in situ PSF work, and you just use your bead PSF to look at the structures deep inside the tissue or cells, what you're going to get is artifacts. Over here, you can see this is the mitochondria imaged through a 90-micron deep specimens. And you will look at the reconstruction in the cross sections over here. You can see different band-like structure. These band-like structure induced by the spherical aberrations is due to a such large index mismatch aberration when you go deeper into the specimen. If you have spherical aberrations, most of your point-spread function will look like circular. And your localization algorithm look at the circular point-spread functions, going to say they're mostly in focus. Multiple Z sections of these mitochondrial structure give you different band-like structure, therefore give you artifacts. Not only artifacts, we can also quantify the information decreasing with the increasing depths. And over here shows in red, you will have slightly decrease in information in the lateral precision with increasing depth. This is only modeling index mismatch. However, your axial resolution will be deteriorating very rapidly. Almost to 20 to 30 microns, you can deteriorate up to 2 times. And therefore, we actually quantify these in theoretical information limit. This is only talking about the precisions, assuming that you already have an algorithm can extract the information in full. So I'm going to talk about 2 different methods, which is one is a numerical retrieval method, the other is physical correction method to allow you to image deep into the specimen. The first one I'm going to talk about requires no hardware difference. It just requires you to familiar with one software and run it, and what we call the in situ point-spread function retrieval. This is a method developed by Donghan and Fan, and two wonderful postdocs in the lab, and they already move on, have their own lab. They developed this method called in situ PSF retrieval. The question is, the PSF calibration using beads is actually inaccurate in vitro, but really in situ point-spread functions what you need for accurate single molecule localization microscopy inside thicker tissues or specimen or whole cells. So the question is, how do we obtain an in situ point-spread function? If you look at the data set we typically obtain in our system, we're actually full of point-spread functions. In fact, our data just point-spread functions after point-spread function, if you look at this. And all these point-spread functions, you can actually segment them and put it into a library of point-spread functions. If you look at these point-spread functions libraries, they can represent a random drawings from the in situ point-spread function or underlying in situ point-spread function you really want to retrieve. However, the link between the point-spread function library you built from your data set, but the in situ one is actually missing. So the question is, if only we can assign each of the emission patterns we get from our single molecule data set to the correct position, then we can directly obtain this underlying truth of the point-spread function description for our point emitters. INSPIRE, or in situ point-spread function retrieval based on blinking data in whole cell and tissue, did exactly that. So starting with single molecule blinking data we can get normally in our imaging sessions in cells or tissues, you can segment these into PSF library. And building from the PSF library, first we generate a template. This template coming from the pupil function, which describes the magnitude and phase of the electric wave in your pupil plane of your microscope. That pupil function can be empty or can be a generic theoretical model, does not have to be representing any truth towards what you really detect on camera, but it gives you an initial reference. This initial reference basically can be used as the initial template to then your point-spread function libraries. Each of the point-spread function will be calculated against these references for their similarity. Similar PSF from the acquired data will be grouped together into different bins in the Z positions, and therefore they can be averaged and summed together. Once you've done that, you have an updated PSF stack. These PSF stack can go on and generate a new pupil function. You can see that this new pupil function using experimental data is slightly closer to the point-spread function that we just generate. Using these new pupil functions, you can generate new reference PSF. And these new reference PSF has now incorporated some of the information from the data. Now you can do this assignment again by similarity, do the average, update another round of the pupil function, and eventually after 5 to 7 iterations, you can get is a final PSF model. We find this converging 5 to 7 iterations give you a fairly robust point-spread function model describing the in situ 3D response directly from the single molecule data set. And then we can use this 3D in situ PSM model to pinpoint single molecule locations through whole cells and tissues. This is demonstrated further in, for example, in the simulated PSF. We will show experimental data demonstrations later. And you can see this biplane simulation point-spread function coming from different axial and lateral positions. And the in situ point-spread function models give you almost identical to the ground truth estimation. And the residuals are nearly zero. If you decompose these pupils into Zernike polynomials, inspired and ground truth Zernike coefficient, they're almost overlapping all the way up to tertiary spherical aberrations. If you look at the point-spread function model here, the ground truth point-spread function and the inspired retrieval point-spread function, they're almost identical except some minor points. We can now take this and try to validate it in the experiment. What we do is we put a deformable mirror in our setup in the pupil plane. And the deformable mirror, we used to use it for adaptive optics. We're changing wavefront, correct aberrations. But here we use it as a calibration or demonstration tool. What we can do is we can introduce some aberrations into the deformable mirror and let the Inspire network look at this data and see whether it can retrieve what we input towards the deformable mirror. Deformable mirror gave you an aberration introduced by introducing different voltage differences into their actuators. So what we see over here is blinking data with distorted point-spread functions, and Inspire operate on that and give you this matrix. So here in this axial x-axis is the deformable mirror input, what we input in there for the experiments. And this is what Inspire retrieved amplitude. You can see this is almost diagonal matrix all the way until tertiary modes. We don't have a very good performance on these high aberration modes. So we can go now a little bit deeper. Okay, this is numerical compensation method. It certainly does not restore the information you already lost, but we can go deeper because we have the accurate point-spread function. Now z equals to 9 to 11 microns over here in mitochondria, and you can see this is a very nice image in lateral, but if you cut cross sections, you can see the comparison between using Inspire and using the current previous state-of-art in vitro PR method. You get sharper contours of the mitochondrial membrane. You also remove the artifacts that come in when you have mismatches between your point-spread function, where you really detect the point-spread function you obtain from the beads. If you look at the in vitro retrieved point-spread function, which is on bead point-spread function, or Inspire one, which is the in situ one, that you can see the difference. This is actually reflecting the spherical aberration you introduce imaging a little bit deeper into the specimen. You can now use this to image brain sections of 30 microns, and these are the immune-labeled amyloid beta fibrils. And these fibrils are dominant in Alzheimer's disease models and brain sections that we use. And the number of localizations around 500,000, XY precision 7 nanometers, and Z precision about 28 nanometers. And these plaques used to be quantified through intensity and size. Now you can see the intricate structure of these fibrils arranged in a three-dimensional manner. If you cut cross-sections around some of these axes, you can actually see them running up and down in a three-dimensional space. All right. However, numerical method can only give you the information up to the limit that you detect. When I talk about information, I'm referring to Fisher information. Due to the photons once detected is already Poisson noise corrupted. The information that are lost due to wavefront distortions are lost permanently. To recover those informations, you have to put a physical element to compensate the wavefront errors before actually detecting on the camera. This requires we to do adaptive optics. Just to go back and talk a little bit about adaptive optics very briefly. For example, over here we have a cell. You have a GIP in the middle next to the ER, and they generate spherical wavefront. And one of the objectives, the most important job, is to converting this spherical wavefront into plane wavefront. And after that, you will put a tube lens, and these tube lens focus this plane wavefront very nicely into a diffraction-limited point-spread function. However, cellular and tissue constituents will have different refractive index. Light propagates through these constituents have different speed. If you have a spherical wavefront, you go through at different materials, and therefore it will give you a distorted wavefront just due to the speed difference of light propagation. And therefore, if you give a distorted wavefront input to the objective will now be able to convert it into a plane wavefront, therefore give you an aberrated wavefront. This aberrated wavefront, after turbulence, give you aberrated point-spread functions. These point-spread functions decreases in their Fisher information content, give you less localization precision, poor resolution. On top of that, it's just very difficult to pinpoint the center of this molecule. For example, look at this very aberrated PSF. Adaptive optics works by putting adaptive element into the pupil plane of the objective, and therefore you can curve the wavefront, curve the element, the reflective membrane surface exactly at the wavefront shape, and therefore correct the wavefront shape. And still, if you put a tube lens over here, you will get a fairly nice diffraction limit point-spread function. Now the question is, how do we know the wavefront? Because we cannot directly measure it in single molecule localization microscopy. You cannot put a guide star in there. It's difficult to measure the wavefront. How do we guess it? So this goes back into one of our projects that we first explored in 2016. What we're just trying to do is try deep learning in single molecule. At that time, Pei just started her PhD. She wanted to do that. So we developed this neural network, which actually is fairly powerful in the end. We developed a single molecule neural network, and based on ResNet 18, we also introduced information-based training, basically treating different single molecule training data set, their information into the cost function. And what we achieve is we can train this network with simulated or experimental PSF, very complex ones. After training, we can actually estimate the point-spread function of very complex point-spread function on multiplex information they contain. From these point-spread function patterns, we can estimate their 3D location, orientation, and wavefront distortions. The SMNet does not only give you an estimate, which is demonstrated over here. What we show over here, this radius of these circles are errors of localization. And we plot these errors across different background count and different photon count levels. And we have two types of circles. One circle is a Kramer-Lower bound, which is the statistical error limit. There's no chance for you to do better than this if you want an unbiased estimator. And SMNet performance is plotted in blue circles. And you can see at different background count and different photon levels, the blue circle and the red circle always overlap. That indicates SMNet extracts the information at the theoretical limit. And we can also do this with biplane and with double helix. And here is the deep learning localized single molecule mitochondria data set, which is of very high quality. However, we move on from there, and to the next iteration of this SMNet, we actually want to use single molecule neural network to drive a device. This deformed mirror is a 140 element device. It's very difficult to drive efficiently. But deep neural network is just a tool to actually doing that complex inference and complex driving performance. We develop a deep neural network driven adaptive optics. Over here, you basically have your data. Your data is segmented into PSF. You can see these are aberrated PSFs. Goes into this basically similar architecture as SMNet. These SMNet, or deep neural networks, give you aberration amplitude estimates for different aberration modes. And each single molecule gives you one estimate. We put them into a Kalman filter, which basically combines the previous history estimates and current predictions, and focus on the high and low uncertainty predictions. And after that, we can decipher what are the experimental mirror modes, what's their amplitude, and how much they are. And then we use these ones to give you a voltage map feedback toward the deformable mirror, and then we compensate the aberration in our system. This allows us to actually compensate the aberration from small to large scale, from 0.75 to 2.75 root mean square wavefront errors in radian. So you have low aberration levels, you can compensate with DLAO. You have high aberration levels, which you definitely don't want to take single molecule data from these type of data set. But after DLAO, you can get a very nice diffraction limit spot restored. And how fast it is. Actually, only with one cycle of iterations, it almost compensates 70% of the aberrations. Here's a demonstration. Here you can see very aberrated point spread functions. You don't want to take data on this sample. But if you run DLAO, after one round of compensation, you can see they are largely restored into diffraction limited spots. Those spots you will feel very confident that give you good reconstruction results. And how robust it is. We really like the Boston Dynamics movies that, for example, you can introduce arbitrary interruptions on those robots. These robots still function pretty well. We did something similar. We actually put a deformable mirror. Well, we use deformable mirror to compensate aberration, but we introduce arbitrary aberration at arbitrary time. We don't tell the DLAO when we did it and how much we did it, and he don't know. So now you can see aberrated point spread function in the middle. The DLAO is just basically trying to drag the aberration down. Okay, now we can see, oh, it's pretty good, so let's kick it. So now you can see we put in some random aberrations, and DLAO look at that and say, this is not what I want. So we keep dragging the aberration down continuously. We can run this for about 3 minutes, and we can kick in aberration every 10 seconds. And this is a demonstration of, I think, 13 or 15 interruptions. And it's running fully autonomously. We don't have to give any initialization command or do anything on our side. So basically, it gives you autonomous AO control. We also tested if you move the specimen during acquisition, it can also compensate the aberration immediately. Without that, we can actually image a lot deeper into the specimen. This is what we image in brain samples. These are the dendrites, and they are a conventional image of the spines. You can already see some of the very nice spines showing up over here. This is diffraction limited, so what you see is similar to what Goji actually said before. If you do super resolution, what you can see that it's much better described in spines. In fact, that you can cut cross sections around the neck and the head. You can see the rings of their membranes. You can also actually calculate the volume. You can see how the neck actually sometimes pinching off. You can identify all the spines much better with much higher precision measurements. And all of this done is 130 micron deep into the brain tissues with 250 micron cut brain tissues over here. You can also do some numerical quantification out of the neck and head size. We also developed a method to quantify the volumes of the spine from the neck to head. We can have an evolving volume with very high precisions. We can also go back to look at our amyloid beta fibrils, and these amyloid beta fibrils are looking very similar in conventional microscopy from A to F over here. However, if you're trying to do super resolution, you find out they are drastically different because now you have such a higher resolution. Some of the amyloid beta flag are actually are actually coming from individual fibrils, and some of the amyloid beta plaques actually are diffusive. So not only that, we can potentially study these plaques, whether they are the size of it or the intensity of it, we can also look at their ultrastructures. At last, given that such strong technology expertise is in this audience, I want to share with you some of the theoretical work. There's no experiment here, just calculations. But I think this will be interesting to some of you. So a revisit to a theory of diffraction limit. We all know Abbey, 150 years ago, proposed this diffraction limit, which still governs almost all imaging development in this, even nowadays, in our modern microscopy world. And basically says structures smaller than delta R cannot be resolved. And the delta R is proportional to lambda and inverse proportional to the NA. And this is actually carved in stone in University of Vienna. And we have a diffraction limit, which basically proportional to the inverse of the NA. The NA corresponding to the angles of the light that you can collect from your objective. Smaller aperture, which indicating smaller NAs, will give you large PSF. And big aperture, or corresponding to big NAs, give you smaller PSF. In our microscopy development, we always want smaller PSF, such that we have higher resolution. However, there's one additional factor that has not been incorporated, that is noise. This is because when Abbey developed the diffraction limit, the most of the microscopy is transmission microscopy. If you lack of photons, you shoot more photons in there. But nowadays, with modern microscopy, most of us are dealing fluorescence. And each single molecule gives you about 500 photons to 1,000 photons. Then they disappear. So we are always photon stuffed. So how does that affect our resolving power? Well, you can see over here, with very little photons, you simply cannot resolve the structure. But with more photons, you can resolve this structure now. So there's one thing missing, is that with objective NA and lambda are fixed, the photon levels really determine the practical resolving power. If you don't have any photon, you cannot resolve anything. Here's another demonstration. You have an object, and your system certainly can visualize this image because it's within their OTF. However, with noise, you can barely see them. So the question is, how do we incorporate photon statistics, noise, into the resolution limit? In other words, Abbey's resolution is a function of NA and lambda. Can we write a new resolution, which is a function of NA, lambda, and photons? Following the Abbey's approach, basically look at different gratings at different frequencies and shrink the aperture and tell about the visibility of the system. Basically, that's how he makes his experimental discoveries. We can do is we can actually also investigate grating structures. But what we incorporate is a Fisher information. We calculate the Fisher information density of all these grating structures at different photon counts and calculate how much Fisher information density on their phase. So the grating phase is very, very interesting. The one phase against another phase basically tells you how much shift you have in your grating. In other words, if you cannot tell whether your phase is cosine or sine, I think you don't really see the structure. So by using that criteria, we can actually generate an information-based resolution. This is purely theoretical. You don't have to build a system. There's no sample is imaged. You just calculate. As you can see, there's some demonstration over here. The diffraction limit is 250 nanometers. Wide field does not achieve the diffraction limit under 5,000 photons per square micron. ASM and SIM both supposed to achieve 125 nanometer, twice of the diffraction limit, but they do not. Sometimes they achieve 220, and sometimes they achieve 170. If you give them infinite photon count, they will achieve 120. But if you have limited photon count, you cannot. In other words, in IBR, you can imagine photons are the information currency for resolving power. And you take these currencies, you can buy resolution. You can buy it from confocal. You can buy it from structural illumination. You can buy it from ASM. And these systems give you different efficiency of using those photons. A demonstration showing over here. Here is the structure 1.85 NA divided by lambda. which is actually resolvable. Okay, it's resolvable in wide field all the way through structural elimination. But if, sorry, one more minute. If you want to use wide field to resolve these structure, you need 100,000 photon per square micron, which is very difficult to achieve. However, if you want to resolve structure in SIM, you will require only 1,000 per square micron, which almost all sample can do. So this is much better efficiency, even if your structure is resolvable in wide field, to use super resolution to boost that efficiency, give you more resolving power. And I will just skip. NA requirement can also be demonstrated. You no longer need oil immersions, but you can use air because you have more resolving efficiencies in super resolution. Pinhole can be evaluated. You can even do more than Nyquist sampling rate, actually give you efficient boost on information density. With that, I'd like to thank the student postdoc who did the work and funding, and thank you very much for your attention.